Here friends, we are going to learn about mutations, which is called sudden change in genes, or any aberration take place or alteration take place in genes or DNA or chromosome is called mutation. Mutation is always caused by agent named mutagens. So mutagens cause mutation, which is sudden change in genes and alteration in DNA, gene and chromosome. As you know, DNA is having genes in it, and chromosomes are formed from in the DNA only, and gene is said to be the smallest unit of heredity. If we see this diagram of the chromosome, this is a chromosome. It's one part of the chromatid of the chromosome. This is the part which is called centromere, and these small parts which are shown by the alphabets A, B, C, D, E, F, G, H, I, J, these are called gene segments. Always chromatids are made up of gene segments and they are given some name. In this diagram we have given alphabets, and in this diagram we have given the numbers to it. These are also gene segments of another chromosome, which is given number 1, 2, 3, 4 and further on we can give it. The change in the gene or any change in the gene is called genetic mutation. Genetic mutation take place in the genes of the chromosomes of the human body. It can take place in any other organisms also. Mutations are caused by factors which causes alteration in it, and that are called mutagens. Mutagens are of different kind. Radiation, example is X-rays, UV rays, chemicals, different kind of chemical like colchicine. If it goes through the nails, it can also lead to the mutation. Spontaneous changes are the mutations, and these are also due to deamination of cytosine to uracil. It may occur due to errors in the replication. It may occur due to errors due to recombination during crossing over in the meiosis cell division, or it is called spontaneous depurination. When we see this is the chromatid of a chromosome, because each chromosome composed of 2 chromatids. Alteration in DNA or gene leads to the shift in the message coded by the DNA, because DNA always read the protein message which has to be coded through the codons. Codons are always made up of 3 base pairs. So it is called Frameshift mutation. When the reading message of another protein is being coded, that is called Frameshift mutation, or the message read is different. Shift in the reading frame leads to genetic mutation. These kind of mutations which are there are of 2 types: point mutation and Frameshift mutation. Point mutation take place at a point or certain part of the base nucleotide. Replacement of one nucleotide by another is called point mutation, and it is of 2 types: transition and transversion. Now we know the purines are of 2 types, adenine and guanine. Pyrimidine are of 2 types, cytosine and thymine. Now further, the point mutation is of 2 types, transition and transversion. In transition, purines or pyrimidine replaced by another. Example, A replaces G, these are purines, or C replaces T, pyrimidines. C is cytosine, T is thymine. Now what is transversion? In the transversion, purine replaced by pyrimidine and vice versa. Example, A replaces T and C replaces G. Now what happens in the Frameshift mutation? Frameshift mutation is of 2 types. One is deletion and another is insertion. Example for deletion is cystic fibrosis. From the name deletion only it is clear, it means some part of the gene or the segment of the DNA is removed or deleted from the chromosome. An example is cystic fibrosis. Insertion, from the name it is clear, something is inserted in the chromosomal arm or the chromatid, or of different chromosome or the same chromosome. Example, thalassemia is the example, which is a disease. Now we come here to the chromosomal mutation. What happens in the chromosomal mutation? As I told you, this is the chromatid with different genes and segments of gene which are given different names. This is the normal chromosome chromatid. When some change takes place in it, it is called disruptive chromosome. Suppose in this chromosome the gene segment names are these only, and if I remove F chromosome, part of the F gene part from this chromosome, it will be leading to disruptive chromosome where the F will not be seen, or it may code for different protein. It may cause some kind of disease, or may not. Disease can be there. Change in structure of chromosomes is called chromosomal aberration, which is of different kinds: deletion, duplication, inversion, insertion, translocation. From the names, the meaning of the word is clear. Chromosomal mutation take place due to disruption in chromosomal structure. Also, disruption in chromosomal number also take place, and it is also chromosomal mutation, which is called numeric chromosomal aberration. Now we will see what are the meanings of this deletion, duplication, inversion, insertion and translocation. Here some diagrams are shown. For example, this is the chromatid of the chromosome. Let us have same name: A, B, C, D, E, F, G, H, I, J. Now the red part of this chromatid is showing gene segment. If this red part of the gene segment is removed, it is written, it is called deletion. Method is called deletion. Here also it is written with the underlined by green color. Now we come to duplication. As from the duplication meaning only, it is known like some copy is being created or duplication of some gene segment take place. Here the red part is shown, which is called the gene segment or the gene. If it is duplicated, the size of this chromatid will increase in size. Duplicated part is shown by red part. Chromosome is showing length change, and it may form some other kind of protein. As you know, the DNA carries the message for formation of certain kind of protein. If any change take place, it may code some other protein, or it may not code some other protein, which is called silent mutation. When the protein is not coded, or change or variation in the protein is not taking place, it's called silent mutation, where the gene change or due to duplication or due to different method is not affected. Now this is the method which is called inversion. From the word inversion only it is clear something is getting inverted, upside down. Now this is one green part of the gene, and this is red part of the gene. The arrow shown here. First it is like this, red part is up and the green part is on the lower side. Now when translocation take place, this two gene segments get inverted. The green which is on the lower side will go upside, and the red one will go downside. So inverted part is shown here in the diagram, second one. What happens in the insertion? From the name only it is clear. The gene segment is inserted in the other chromosome. This is the first chromosome A, and this is the second chromosome B. Now what happened during the insertion? The red part, which is bordered by the green part, green lines, when it goes into the B chromosome, the red part is inserted here. The red part is inserted in from A chromosome to B. So this is called segment from the other chromosome is inserted into the second chromosome. Now what happens in the translocation process? Translocation meaning only it is clear from the word transfer plus located. When segment of one chromosome is translocated on the other place on other chromosome, it is called translocated. This is chromosome A and this is chromosome C. So when this gene segment is translocated to the other chromosome of the other chromatid, it is called translocated segment. What is the difference between the base substitution or point mutation? Base substitution and point mutation means one and same thing. Point mutation occur when only one base in DNA is altered. Single base change can be transition type or transversion type. Transition in which one purine is replaced by another purine or one pyrimidine is replaced by another pyrimidine. Transversion is a process in which a purine is replaced by a pyrimidine or a pyrimidine is replaced by a purine. Friends, if you want to upload any other video of any other topic in science, kindly let me know. Everyone study again to recall things in your mind. Everybody can do anything if you decide to do it and to learn it. Everything is possible. Kindly like it. Also subscribe it. Thank you very much.